I am Ms. Vaishnavi from Triple College for Women from second MSc Biochemistry. And here I am going to talk about the topic Eukaryotic replication. The replication is the process of conversion of parent DNA into the two daughter DNAs. This parent DNA acts as a template. Cell cycle consists of 4 phases: G1, G2, S phase, M phase. G1 phase prepares the cell to divide. S phase copies all the DNA to form a new daughter DNA, that is replication. G2 phase, the genetic material is condensed and the cell prepares to divide. M phase results in the formation of 2 cells. Semi-conservative replication. This produces 2 copies of DNA in which one contains one of the original strands of DNA and one new strand. The enzymes and proteins involved in eukaryotic replication are RNA primase, DNA topoisomerase, DNA polymerase alpha, beta, gamma, delta, and epsilon. pCNA, RPA, RFC, ORC, MCM, and DNA ligase. Formation of pre-RC. The pre-replication complex is the complex structure composed of 6 ORC proteins CDC6, CDT1, and MCM. This takes place in G1 phase of the cell cycle. The first step in the assembly of pre-RC is the original replication complex binds for the origin of replication in the DNA, which is AT rich. The ORC CDC6 complex forms a ring-shaped structure. The CDT1 protein is required for the licensing of chromatin for DNA replication. This facilitates the loading of MCM2 to 7 complex onto the chromosome by stabilizing the left-handed open ring structure. CDT1 is associated with the C-terminus of CDC6 to promote MCM binding. This is pre-RC, pre-replication complex. The replication involves 3 stages: initiation, elongation, and termination. Initiation. The pre-RC enters the S phase from a G1 phase by activation. At the transition of a G1 phase to S phase of the cell cycle, CDK and DDK transform the pre-RC into an active replication form. CDK phosphorylates the CDC6 and activates it. DDK phosphorylates the MCM complex and activates it, and CDT1 protein is inactivated by the germinin protein. And in addition to MCM binding, CDC45 protein is also associated with the MZM, which helps the loading of DNA polymerase alpha, epsilon, RPA, MCM, and pCNA onto your chromatin. The ORC is inactivated through phosphorylation. The genes complex is involved, which is a complex structure made up of 4 proteins: CDC 105, 103, 102, and 101. This helps in the interaction between CDC 45 and CDC and MCM. This CDC, MCM, and genes complex forms the CMG helicase, that is replicative helicase of the replisome. This is a diagrammatic representation of the formation of the ERC, and here the degradation of CD6 and inhibition by germinin is represented, and there's results in the formation of CMG helicase. And elongation. The primers are added onto the leading strand and the lagging strand, and few nucleotides are added by the polymerase alpha. Coming to the elongation, the RNA polymers are added on both the strands. In the leading strand, the RNA primase placed in 5' and to 3' direction, and in the lagging strand, many RNA primers are placed. The RPA proteins are placed on the strands to prevent reannealing, and the DNA polymerase alpha adds few nucleotides for initiation. The pCNA is loaded into the DNA by RFC. then loads the DNA polymerase delta. This works in the 5' end to 3' end and adds nucleotide only to the 3' end. The replication is continuous in leading strands and the discontinuous in lagging strands. In the lagging strands, the discontinuous stretches formed and are called as Okazaki fragments. The topoisomerase 1 and 2 relieve stress and prevents supercoiling. This DNA polymerase delta creates a flap by the displacement of primer, and this flap is replaced by FEN1 flap endonuclease 1. This flap is actually removed. And then RNase H enzyme recognizes the RNA DNA hybrids and removes these from the replicated strands, leaving behind primer template junction. The DNA polymerase alpha recognizes these sites and elongates the breaks, and DNA ligase 1 repairs the nicks that are left between 3' OH and 5' phosphate and binds it. This is called as Okazaki fragment maturation, and CTF1 and 1 proteins are also involved. Claspin 1 is highly involved for the efficient replication fork. This is a diagrammatic representation of the elongation. Termination. Eukaryotic linear DNA has multiple origins of replication, and one fork reaches end and encounters another fork. SCF E3 ubiquitin ligase complex catalyzes the MCM7 by the addition of ubiquitin. This polyubiquitinated MCM7 is recognized by the protein P97 or valine containing protein. This process is called as K48 ubiquitination or molecular kiss of death. This results in the detachment of CMG-SMD and terminates the replication. This is the diagrammatic representation in which one fork counters the another, and this is the recognition by the P97 or valine containing protein, which actually terminates the replication. Thank you.